It is a more general question, about specifically about also about Stephen Archer's saturation experiment. The selectivity for the substitute particle, grazing particle, is specificity for or selectivity does not matter, right? Because all you're trying to do is saturate it.
Yeah, that's right. That's,
yeah.
In theory, as long as they spend some time processing the prey.
Exactly, as long as there's handling time associated, yeah. The more general comment is we're always looking more in the surface ocean about the relationship of grazing and microzooplankton. But obviously the protists occur in great depths. And what we found is what Debbie suggested, that we see some lower thresholds of prey abundances below which grazers cannot. So essentially the prey has some refuge.
Right. Yeah, that's interesting. I'm not sure how it works in that respect.
Except for grazing on particles.
Right.
Is that my next? Yeah, I have a question for Ben. Right here, sorry. That number you threw out, about 80% of the plankton are mixotrophic. Can you elaborate? Are we talking about 80% of the species? It certainly can't be 80% of the biomass. It can't even be close.
No, that was in Mike Zubkov's paper. That was a particular result there. I think 80% of the pigmented biomass was
of the biomass. But how could that be when Synechococcus and Prochlorococcus are the most dominant plant life form on, one of the most dominant life forms on Earth? I'm not aware that they're mixotrophic.
Were they just talking about protist biomass?
Yes, I think they were. Thank you. So yeah, I think that was, Ginny's just suggested it was protist biomass. I can't remember, but I know that Prochlorococcus, Synechococcus are numerically abundant, but not necessarily in terms of biomass.
Well, they are a huge amount of the plant biomass on Earth.
They are, yeah.
Yes.
But there's the larger protists are probably a larger fraction of the total mass, I would think.
Yeah, I don't know.
I'm trying to remember that paper, and I think it was like the flagellate
okay.
biomass.
Susan, she has this.
I can try without the microphone. Hopefully not mine. Thank you. And thank you for highlighting the rich chemical and behavioral and phylogenetic diversity in protists. And I was just going to stimulate that we think collectively, also including with the presenters this morning, about how do we incorporate this diversity adequately in models, and conversely, how can empiricists deliver data that is useful for models without oversimplifying. I was just thinking, Ben, during your talk, I can think of large ciliates that are, they live the fast and hard lifestyle, and the same sized heterotrophic dinoflagellates that are gleaners. So size, is not the distinguishing feature there. That's not the driver. And of course, I know you have to be simplifying in the model, but how do we incorporate complexity without making a 900 million box model?
It's something I've thought about a lot, and I'm someone who likes to generalize, because learning lots of taxonomic groups was never my strength when I was doing my undergrad biology degree. But so I think we can have a dialogue between different communities where some people try and simplify, and modelers can do that, but they've got to realize that they are modeling complex systems and not forget that. So keep questioning the simplifications, keep looking at the biological observations and try and find where your generalizations are failing, and then look back at the observations to see where you can improve your models.
I think, to chime in there, I think there's some information is going to have to be incorporated about the relative abundance of different classes of organisms, and some empirical laboratory experiments on the grazing characteristics of those major groups, and then perhaps some comparison of those data to estimates of grazing produced by, methods that we've heard about, or perhaps in situ approaches, where you don't even bring a water sample up from the water column, just to compare, to see whether you can parameterize, and then maybe you can come up with a general bookend, if you might, for different types of water columns, because the answer is not going to be the same whether you're talking about the surface ocean, whether you're talking about the mesopelagic, whether you're talking about a stratified water column or the deep ocean. So.
Yeah. I think you shouldn't shy away from that complexity. You should be building these complex models, but then using them as a way to draw out the really important, the primary sort of processes that you can then incorporate it in bigger biogeochemical models. That's an approach taken with a lot of different aspects of biogeochemistry.
So I'm going to come back to the syndineals because my group's been doing 18S data sets, and you're absolutely right, they dominate these sequence libraries. And so I'm just trying to think about, is it a consequence of maybe something that we're missing, especially in the molecular ways that we're doing taxonomic composition, and that maybe it's because they have incredibly high 18S copies? Because when we look down a microscope, I mean, is it also that we wouldn't necessarily notice what they look like? Do you have any comments in relation to that?
They have some characteristic autofluorescence that comes in very handy. And so counts that Taylor's doing, for instance, in Salt Pond, they're not insignificant. Okay, so I totally agree with you that there's going to be bias when you're looking at a gene like ribosomal RNA for any alveolar group. So that's almost invariably overestimating, their true abundance. So you can't equate those gene copy numbers to cell numbers. However, you can compare abundances of those copy numbers from region to region for the same taxa and get some useful information. And you can couple that with microscopic counts of dinosaurs, for instance, which are fairly easy to do. We're doing them in the lab now. And I was surprised to find that 35% of the population in Salt Pond, which is a fairly, it's not totally eutrophied, but it's organic, rich, coastal, marine pond right here down on Surf Drive, is infected. I think that's fairly significant. So we want to get infection rates at the MVCO observatory as well, out in more open ocean site, and some locations off the coast of France as part of her thesis to get some ideas about that.
I have a question, actually, over here. I have a question for Ben, and maybe a more general question. So you were talking about mixotrophy in terms of phagotrophy. What about osmotrophy and uptake of dissolved organics?
So we make that distinction, firstly, because I think everything is probably going to show osmotrophy. So yeah, it's something we need to consider. But I've drawn a line there so I can focus on phagotrophy, and other authors have made the argument that that's a distinction, firstly because it restricts the number of species you're looking at, but also it has a much more direct impact on the food web when you're killing organisms directly rather than just eating the detritus or taking up organic compounds. So osmotrophy is, I think a very prevalent process, but it's not something I've focused on.
The question is, you were talking about surrogate prey, and have you looked at the different sizes of surrogate prey and see what the influence that would be?
No, but
And especially relative to the predator size.
I think that's it's a really good point, and you can use that as a tool, to get a grazing pressure on different size classes. So absolutely, but we haven't done it yet. But yeah, it's a good idea.
One thing that you can try perhaps is labeling, heat killing and labeling your in situ population of prey, and then providing that as your surrogate prey. We did that once and got some interesting information about what type of prey were being eaten.
Should be on. Okay. I had a question for you, Jenny. I'm just so curious. I was happy to see you show the oxygen data. I'm curious to know how these guys persist. If you knock out methanogenesis chemically, they don't do well. But if you knock it out by raising oxygen tension, the ciliate does fine. So I'd like to hear your thoughts on that.
We're trying to figure that out, but I think the methanogens get some protection from oxygen and reactive oxygen species from their host, okay? So at least those types of genes are being expressed that would handle that. So I'm not sure, we haven't gone over 4% oxygen, so I imagine it may not hold up, but methanogenesis is still going on at those low oxygen levels.
I had two questions. One's for Ben. How come I don't have chlorophyll? Or, is there a cutoff as you get larger that having mixotrophy doesn't benefit? And the other question was just the aspect of, do all these grazers ultimately increase primary productivity of systems, mostly by, recycling nutrients, so that without them we'd actually see lower productivity?
I think you're probably too big to get enough water to photosynthesize effectively without growing roots. So yeah, there is a size limit. Then I guess it comes down to there's a surface area to volume ratio for one thing, and just getting nutrients as well. But I think the biggest I've seen photoheterotrophic wasps, which have pigments on their heads, so they have sort of foraging behavior that's around about the noon.
Some jellyfish that have cigars. They're big ones.
Yep. And does grazing produce primary production? I think it can increase carbon export, and it can increase primary production in larger size classes. So then you see like carbon enrichment in those size classes, and they're also sinking deeper, so it's effects through that. They're not. I think they can increase primary production, yeah, but it's still ultimately limited by the nutrient supply on the system as a whole in terms of the nitrogen or nutrient cycling. But maybe you can get a bit more carbon in there, a bit more carbon efficiency. Maybe is the best way of putting it.
A long time ago, Ralph Lewin wrote a comment called, "Why Aren't Cows Green?" It was quite amusing, and his conclusion was that cows aren't green because they're big and they're not translucent, so you could do it if you're a jellyfish, but otherwise, there's no space. If it was outside your body, you couldn't use it. If it was inside, it wouldn't get any light.
Sounds fair enough.
Well, we answered that question. We can thank the panel.